Imagine watching a movie where the soundtrack is half a second ahead of the picture. You'd notice instantly, right? Now, picture the same delay happening inside your own eyes. Yet you never notice a thing. How does the brain pull off this seamless magic trick every waking moment? That question has just cracked open a brand new chapter in neuroscience. And the answer is hiding in the last place most experts expected: the retina itself. A jaw-dropping study led by scientists at the Institute of Molecular and Clinical Ophthalmology Basel, IOB, the University of Basel, and ETH Zurich reveals that your retina doesn't simply pass raw camera feed data to the brain. Instead, it acts like a miniature timing engineer, actively recalibrating the arrival times of visual messages so that everything feels perfectly in sync, even though the cables carrying those messages are wildly different lengths. But here's where it gets controversial. The team suggests this built-in retinal clock might be more important for everyday vision than the famous myelination strategy the rest of the brain relies on. Yes, you read that correctly. The eye may be outsmarting the brain at its own game. Let's unpack how they discovered this and why it could force textbooks to be rewritten. The anatomy that started it all. At the center of your vision lies the fovea, a pinhead-sized pit packed with photoreceptors that give you razor-sharp detail and color. Surrounding it are millions of other photoreceptors, whose signals must exit the eye via nerve fibers called axons. Here's the catch. Axons are forbidden from crossing the fovea, because even a tiny scar would blur your precious central vision. The workaround? Axons bend around the fovea like cars on a racetrack, creating a detour that can be several millimeters longer than a straight-line route. Longer cable, slower signal. Simple physics, right? Not so fast. The researchers noticed that reaction times across the fovea were eerily uniform. Something had to be compensating for those extra millimeters. From donated eyeballs to living retinas. Eyeballs to living retinas. To solve the puzzle, the team received entire human eyeballs from organ donors and kept the retinas alive and firing. No small feat. Using high-density microelectrode arrays that record electrical fields 20,000 times per second, they filmed the departure lounge where signals leave the fovea. They then measured how fast each signal traveled. Next came nanoscale detective work. Transmission electron microscopy let them measure axon diameters down to the nanometer, while fluorescent labeling and high-resolution microscopy mapped every twist and turn of the wiring across the whole eyeball. Finally, they built a mathematical model that linked axon length, thickness, and conduction speed. The compensatory trick nobody predicted. The model delivered a crystal-clear pattern. The longer an axon, the thicker it becomes. Thicker axons conduct electricity faster, exactly offsetting the extra distance. In other words, your retina preloads speed boosts so that every signal arrives at the brain's visual cortex at the same moment, like staggered runners given head starts so they all cross the finish line together. To prove this isn't just neat anatomy, but has real-world consequences, the team turned to adaptive optics scanning laser ophthalmoscopy, AOSLO. Why this flips conventional wisdom on its head? Most neuroscience courses teach that myelination, an insulating fat sleeve around nerve fibers, is the brain's primary tool for speeding up and synchronizing signals. Yet retinal axons are naked. They have zero myelin. If the retina can synchronize without it, then unmyelinated pathways elsewhere in the nervous system might be doing the same thing. That's a direct challenge to decades of doctrine. And this is the part most people miss. The retina isn't just a passive sensor. It's an active preprocessor, fine-tuning temporal precision before the brain even gets involved. Some vision scientists argue this makes the retina more intelligent than we ever gave it credit for. From lab bench to hospital bed. Understanding this mechanism isn't academic trivia. In diseases like glaucoma, the longest retinal ganglion cell axons are the first to die, precisely the ones that need the most energy and mechanical support. If doctors can learn how the retina keeps these marathon fibers in sync, they might discover new ways to protect them. The team's next mission is to model what happens when the timing system breaks down. They also want to know how the retina knows, which axons need to be thicker, and how this calibration develops from infancy to adulthood. Could similar tricks be hiding in the auditory or somatosensory systems? The researchers are betting yes. Your turn to weigh in. So, is the retina the unsung hero of sensory synchronization, quietly outperforming the brain's own myelination strategy? Or is this just one piece of a larger puzzle we've yet to assemble? Drop your thoughts below. Agree? Disagree? Or share a wild theory? After all, science moves fastest when ideas collide. And if you're feeling generous, consider supporting independent science journalism, so stories like this keep reaching curious minds everywhere.